Hello. So we are Lucie and Blanche, and we are students in Switzerland. She studies at ETH Zurich, and I study at EPFL, and we are visiting researchers here at Harvard. And we are biomedical engineers, students in biomedical engineering. And today for this hackathon, we went a bit out of our comfort zone, tackling a bit futuristic issues, let's say. Our starting point was that aging is a very complex process that involves a lot of pathways, but it has been shown in recent research that DNA damage and epigenetic induce together aging in a non-linear fashion. We'll come back to this later. But epigenetics regulate the expression of genes associated with some cellular processes, and these epigenetic modifications can accumulate over time, resulting in increased DNA damage and dysregulation of cellular functions. And so there exists a positive feedback loop that perpetuates further epigenetic changes, potentially accelerating aging-related decline. Another way of seeing this is that any cellular operation generates a byproduct, the DNA damage. It accumulates over time, which results in aging. And with this vision, the maximum damage that a cell can bear until senescence is the maximal number of operations it experiences throughout time.
So, as we said, these mechanisms of biological aging are very complex. So we tried to simplify it as much as we could to think about it.
Spacebar. Oh, you have to.
Yeah. Okay. So if we make the assumption that a cell is a semi-closed system, and we use the conservation law of thermodynamics, it tells us that the effect of epigenetic modifications and DNA damage can average out. So with this big simplification, we can consider only epigenetic modifications as a predictor for aging. We are well aware that we're focusing only on epigenetic modifications. It doesn't capture the full picture of aging, but we do this for the purpose of our coming modelization. So if we mimic intuitively the way the model of epigenetic modification accumulation would look like, it would be a sum of these two functions resulting in this one. So the first one, f of t, is the predetermined accumulation of epigenetic changes over the course of cellular operations. It's an exponential function, and the factor a in front of it would be the amount of epigenetic modifications that you have at birth from your grandparents and so on. And then the second function, so d of t, corresponds to the environmental triggered accumulation of epigenetic changes with age. It is also nonlinear, and it adds oscillations to the exponential signal. So as you can see, the combination of these two functions is fluctuating. It's not stable over time. Things of the environment like exercise, smoking, sleep, light therapy, and so on can create these fluctuations.
So, as Lucie said, clearly the epigenetic function epi is not linear, unlike chronological age that increases linearly over time. So the goal of our analysis would be to understand the effect of the environment on these epigenetic changes. Epigenetic changes, they induce aging in a way that is not measurable in physical time, because cells, they don't perceive time the way our brain does. The cells don't speak the same language as we do in a way when expressing their age. So we try to understand how we can decode this language to understand our biological age. So instead of observing the age from the external world, we want to observe it from the inside. So we change the coordinate system in which we evaluate time.
Does this remind you of something? So it would be the general theory of relativity. We want to investigate the relativistic correction of biological time. We want to apply the principles of general relativity to formalize how cells age and to decode the perception that they have of their own age. We are going to call this the biological relativity. So we want to study the dynamic of biological processes in cells and not the cells as objects. Thus, we have to introduce a new space where the processes can be represented as locations in the space. So we want to study the variations of the biological rates. For example, if we take the case of methylation, one type of epigenetic modification, this biological rate is the number of methylations with respect to time.
So you can imagine this rate as a massless point along a trajectory, and the trajectory would represent the variations of this rate from a higher rate to a lower rate of methylation changes. And the manifold is all of the different rates it can possibly take. In other words, it's a mathematically defined space where we can represent biodynamics with trajectories. So, we modeled a transformation between a steady biological process and a moving biological process. We can use analogy of the Lorentz factor, for example, where Vm is the speed at which the rate of the biological process changes. So, for example, if we go back to the example of the methylation, it can be the speed at which the methylation rate occurs. That we can call dm over dt prime here. And c, which can be an analog to the speed of light, is the upper bound of the speed at which this rate can change. And with this factor, we can express the time measured from the biological process in motion with respect to the physical time with the relation dt prime equal gamma dt. And so then we just need to integrate this over dt, and it gives us t prime, which is the time elapsed in the accelerated frame of reference in terms of the time elapsed in the physical frame of reference.
So coming back to our example, if the speed of the biological process increases, it means that there is an acceleration, so the t prime is dilated by the factor gamma, like a moving clock that goes slower. And if there is a deceleration, there is a contraction of the time by this calculated factor gamma. In our context of understanding the biological age with the methylation rates changes, t prime is the time for the frame, from the frame of the methylation rate expressed in terms of t, the time that we talk about in our language.
So then if we generalize, like here we only talked about one single biological process, but there are many that are like in our cells happening. So to generalize, our manifold here represents a single biological process, but to understand how they work together, we would need to get rid of the semi-closed system assumption and instantiate one manifold per biological process involved in aging. And then average together all these t prime, all these t prime, to obtain the global t prime. So in this ideal world, we'll find very easily this t prime, and it will be time to test whether our model actually works. And to do this, we'll compare its prediction of the biological time with those of a deep learning model that has been described in the paper here. And it predicts the biological age by analyzing DNA methylation patterns. So if our model results match the age prediction of this deep learning model, then it would be a strong evidence that our concept of biological relativity is working.
So to open a bit, and yeah, we had a lot of open questions. If we manage to prove that this model actually predicts the biological age using DNA methylation, it will help us. I mean, it would help us to understand how aging works better and how external factors influence the way the cells age and so on. But also, we have a lot of remaining questions. Like, for example, it has been told in a talk that different organs in the body have different biological clocks. Intuitively it makes sense because, for example, the skin is more exposed to external factors, so then the functions that we showed at the beginning will have different rates. So then we need to consider different manifolds for each organ because they have different predetermined aging possibly and different curves for the environmental changes. And then if we calculate one age per organ, we also need to understand how to average them because maybe different organs don't have the same weight in the total biological age. And yeah, this is the formula that is put here. And then another question would be the age reversal. If we find the mathematical model, we prove its bijection, then maybe we could prove that biological reversal, aging reversal is possible. And the last one is the age perception. If you understand better how the aging of your body and your different organs age, maybe are you convinced that you age in a different way and then it acts on your actual aging, and this could be explored as well. Thank you for listening.
Awesome. Any judge questions? Quick. Kurt, go.
So you said in the beginning something about general relativity, but then I only saw equations for special relativity. And I'm wondering if that was just like a slip, or do you plan on generalizing it?
That was a slip.
Yeah.
We can think about it more.
Okay.
Question, last one. Nick,
Sure. So, I understand at a high level the distinction between biological and chronological age, but I think the utility of a biological age metric is only insofar as it predicts death better than a chronological age metric at a systems level. So what do you think about that? Because right now I feel like the way we tend to measure biological age, like if you are 80 years old and you get a biological age from some company at age, say, 50, does that mean you're expected to live another 30 years to 110? I would argue probably not. So what do you think about the utility of biological age given current restraints on actually using that to predict mortality and health outcomes?
I think we are not, like the main goal of switching the perspective is not to understand when we're going to die, but it's more to understand how the different like environmental factors that we mentioned, diet, exercise, and so on, have an impact on this aging process. Because we don't see it from the right frame in our point of view. But it's not to predict when death happens.
Okay. Thanks. Any last question? That's it?
All right, give it a hand. Let's help everybody.